start the recording on my side. Good. And then you can start on your side as well. Because basically, in case the quality is not good, I think it's nice to have both recordings. I also noticed that you always have both speakers there. So it's always awkward to sort of watch you nod. Have you considered cropping yourself out after you ask your questions or changing the format?
Okay, yeah, this will be great.
I always found it distracting when I watched your interviews to sort of watch you sort of respond, like not.
Okay, yeah, this is, I think, a good review.
Yeah. So have you thought about sort of cropping and then switching speakers? Like, because, you know, it's fine too to sort of have that.
Yes, so at the beginning I was trying.
Yeah.
Yes, no, you're right. So at the beginning I was trying, but the problem was the resolution. So in some, like if some speakers, they don't have good camera, then it just blurs out. So then I just started using this format and then it just stayed on.
All right.
But maybe today we can go with the speaker mode. So
we can decide after. So I would say let's get started, dive right in. So
yeah. Just a minute. Okay, and I will add your introduction later to the video.
Yeah, you can introduce me now if you want.
Okay.
First, tell me a little bit about yourself, Jitender. So, you know, how did you start this whole thing?
Yeah, so first of all, I mean, I'm close to finish my PhD. So looking for my question, B question. I don't know whether it really makes sense or not to look for B questions like for your postdocs.
Yeah.
But yeah, that's what I thought that I needed. So I just started doing some conversations, and then it happened that okay, I like doing it. So
I'm so glad that you're doing this. I think we need more of these types of conversations, and I think
exactly
it's a good service that you're doing to the world. So thank you for doing that.
Yeah. So now, like the way I start the series, I also started discussing with some physicists now, especially the astrophysicists. So to talk about what is happening in the cosmos.
Yeah.
Like, you know, start from there, talk about then origin of life.
Yeah.
And then I wanted to start something where I talk about mainly about cellular biology. So this is what. So y'all will be the first conversation, you know, in that series. And then I talk about evolution of life, which gives the diversity. And then the last point is the neuroscience. How. So that's like my kind of mini sequence that I'm trying to cover.
I'm excited.
Yeah.
All right.
Okay. So I think I missed.
Do you want me to just answer?
Okay, maybe I will. Yeah, I was just trying to change the view, but I think like to make it speaker view, but it's not.
No need for that. You can always do it afterwards. Like there are, you know, Adobe Premiere and other tools allow you to do that.
Yeah, that's what I use. But okay, anyways, let's start then.
Good.
Hi, Manolis. Welcome to the podcast.
Hey, Jitender. It's a pleasure to be here.
Yeah, so let's start with your journey to become an MIT professor.
So
how did it start?
I was very lucky. I was at the right place at the right time. So I basically have always been fascinated with biology. I've always loved biology and the natural world. It's hard not to be in awe of the world around us. And I hope I'm conveying this to my kids as well, who are basically constantly studying nature and studying life. And they both want to be scientists, like the two older ones. And they all want to study animals, interestingly. So they don't care so much about the human body. I hope that will eventually happen, that they're interested in the human body and the human brain. But my own interests started out very similar to them, basically the diversity of life. I grew up by the countryside in Greece. We had a home in Athens, but most of my memories are from the home in our summer house, basically on the Aegean seafront. and we had a large garden. I would always just play in the garden and run around and sort of, just observe animals and plants. And I had a mask, so I would always dive and go on these long snorkel trips and always observe the incredible diversity of life. So I've always been fascinated with life itself. And when I was an undergrad, I basically studied computer science and mathematics, and sort of the more quantitative sciences is what drew me for most of my life. And then I did my undergrad in computer science at MIT, and I started studying artificial intelligence and sort of how do we create intelligence. And then I was actually taking Marvin Minsky's class on the Society of Mind. I had to write a final paper for that class, and I debated between sort of evolution of language and sort of human thought and how that has shaped language, and evolution of life. And in the end, I basically decided to write about evolution of life. And sort of the problem that puzzled me was how you get complexity, like this exponential increase in complexity that we see, that it basically took, a billion years or 2 billion years to sort of get to life, to the simplest life forms like bacteria. And then, enormous amounts of time to get to the first multicellular life. And then, sort of things kind of sped up. Basically, if you look at how little time has elapsed between this incredible, emergence of complexity. So my very theoretical paper was about the fact that there needed to be another form of information encoded in genomes, which would be about evolvability. That basically maybe what we got in the first billion years or so was to get better at evolving. And sort of that has to do with modularity, that has to do with some kind of reversibility, where you can sort of go back and forth between different environmental traits, where you sort of maintain diversity, where if you look at sexual reproduction, for example, the emergence of sexual reproduction is a major step. But what it allows you to do is now have this ability to sort of have allelic diversity, and by being diploid to sort of have recessive traits that can then sort of maintain some form of memory, if you wish, of ancestral alleles that can be beneficial as the environment is shifting back and forth. So anyway, my paper was very theoretical about sort of evolvability and sort of that as a necessity for genetic algorithms to eventually be able to achieve complexity levels in the billions of nucleotides that the human genome has. And to write this paper, I basically started reading a lot of genetics books on, sort of molecular biology and cellular biology, and sort of trying to understand what would be the instantiation of these constructs. And that basically got me hooked into sort of learning more about biology. And that was the time also that I was basically finishing up my undergrad and master's, and I was getting ready to embark on a PhD. And even though my PhD was in computer science, I ended up writing a thesis on genomics. So I basically did a thesis on comparative genomics of yeast species. And that's when I first started embracing biology as a quantitative, information-based discipline. Like basically the way that I had envisioned biology when I was starting my undergraduate was that you had to memorize a gazillion things about sort of, different parts of life. And as a grad student, the way that I discovered it was more that, no, instead there's a common unifying principle centered on the genome. And that if you could understand the information in DNA, you would basically have a very sort of foundational view of biology and life and biological processes and evolution. So in a way, my PhD project blended my passions for understanding evolution and for quantitative sciences and computer science and information-based understanding of life, and just the foundational principles of the rules of life, the rules of biology, as a set of foundations upon which you could then start layering in knowledge about gene regulation, about the encoding of the genes themselves, and so on and so forth. So that's how I got into this whole discipline. And how I became a professor is totally serendipitous. Basically, Pavel Pevsner contacted me because his student, Ben Raphael, was visiting MIT and heard me at a seminar present my thesis or present some work that I was doing for my thesis. And Ben emails Pavel saying, hey, this guy's great, you should hire him. Pavel emails me saying, hey, you should interview for UCSD as a professor. And I'm a third-year grad student. I'm like, professor? Like, I never actually thought of having a career. I just loved doing research, and I never actually planned anything out. So it's now January 2003. And, 2 months later, my thesis is basically accepted to Nature as a 14-page paper with only 5 authors, and I was the first. And I suddenly sort of come to my advisor, who basically says, congratulations, your paper was accepted. Won't that make a great chapter for your thesis? And I say, well, don't you mean won't that make a great thesis? And he says, oh, yes, that's exactly what I meant. And I'm like, all right, I turn around the corner. I'm like, yes, I can graduate. So I basically had to do my minor. I had to find a committee for my thesis. I had to do a proposal. I had to do my qualifiers, etc. But I had 2 months to graduate. So then I graduated. And my next paper the following year was about the genome duplication of yeast. So again, compared to genomics, but now more about sort of what's different than what's conserved. So basically the PhD thesis itself was about sort of how to read information through the lens of evolution. So basically how to use evolutionary signatures to understand the content of DNA and the content of genomes. And basically my second paper was about the incredible event of a whole genome duplication in yeast that basically gave rise to modern, baker's yeast, the most abundant species of yeast in the fields of Tuscany, and also, in the making of beer and, wine and bread, is in fact the descendant of a whole genome duplication. And basically, for the first time, we could stare across this incredible boundary of a pre-duplication relative and a post-duplication relative that were basically very close and just flanking that incredible event. So suddenly, I could be the first to sort of look at how genomes evolve both during a genome duplication and after genome duplication. But anyway, 1 year later, in January 2004, I emailed back to Pavel, having not responded for a year, saying, dear Pavel, thank you so much for your email. I would love to come visit. So suddenly I was ready to apply for faculty jobs now that my second Nature paper was accepted. And then I went on the job circuit and I got, wonderful offers from wonderful places, including Berkeley and UCSD and MIT. And I basically decided to stay at MIT as a professor. So the challenge, however, is that I did my undergrad, my master's, my PhD, and my postdoc all at MIT. And suddenly I was among the faculty. So I feel that I didn't get the chance to kind of quote unquote grow up and, sort of think of myself as a professor. I still think of myself as sort of the merry-go-lucky undergrad who's just so lucky to be where I am and to constantly learn about so many different things, surrounded by incredibly smart colleagues and friends. And that's basically what has defined both my career and my attitude towards life. Like basically the, just very lucky kid who was basically at the right place at the right time, had an amazing project and amazing opportunities. And I try to sort of give the same thing to my students now, just basically create an awesome environment for research where the sky and their imagination is the limit. And I provide everything they need to do awesome research, go out of my way, write a bunch of grants, gather these amazing data sets, and just give them the opportunity to just explore and go wild, the same opportunity that I was given as a student with just an incredibly wealthy data set, where, whatever I could mine from that data was, news to the world. And I feel that our field has not only not shrunk, but it has exploded. And there's just so many opportunities now for young, smart students in computer science to make a true impact in the biological disciplines, and to truly learn about life, learn about the basic foundations of what makes us human, what makes our brain function, what makes our heart and lungs and immune system work, and also fail in disease. So how do we understand the system deviations from normal behavior or from typical behavior that give us both the incredible diversity and awesomeness of the human body and the human brain in how it varies between individuals, but also gives us the tragic onset of disease, whether it's age-related disorders or whether it's genetic disorders that manifest very early in life. How do we now understand that system, and how do we use the principles of engineering to address the deviations and, when necessary, intervene and reverse the circuits that lead to human disease? So how do we systematically understand the information in the human genome, and how do we intervene and reverse these circuits that go wrong, quote unquote, when disease manifests?
This is fascinating. I mean, fascinating journey, fascinating experience to hear about. And I think just from the intro itself, I have so many questions. um, so maybe first we can define the information itself, because this is a concept which is now kind of dominating in biology, that life is information. I remember reading Paul Nurse's book, What is Life?, where he talks about one of the crucial elements of life is information. So what is this information that we are talking about? And is it really, is it really the best way to look at it?
So it's very hard to decouple the physical instantiation of life from the informational content of life. So it's very tempting as a computer scientist to basically say, oh, I've sequenced the genome of that species, I'm done, I know how to recreate it. But life never recreates itself purely from the genome. Yes, the genome is fundamental and foundational, and it's the blueprint for making more of yourself, but you don't start from scratch nowadays. We are 3 billion years in, and every new life begins with a previous life. So the mother cell, the egg, has so much more information than what's encoded in the DNA itself. And if you look at mammals, it's not just an egg in isolation, it's an egg fed and nourished and constantly interacting with the immune system, with the blood supply, with the nutritional system of the mother, that is basically channeling it as it grows. So when we start thinking about life as information, you also have to distinguish also what information non-life also contains. So, you can think about maybe the number of equations that would be needed to describe a black hole, or to describe a solar system, or to describe a geologically active but life-wise dead planet like Mars, for example. And how many equations would you need to describe all of Mars? And it's easy to sort of simplify and say, oh yeah, there's just a few equations of gravity, etc. But then you have all of the tectonic movements, all of the geological activity, all of the solar winds, and all of the chaotic weather patterns. And at some point you realize that maybe the distinction is blurry between sort of inert and alive objects in our galaxy and in our solar system and in the cosmos. But there's something fundamentally very different about life, the way that we know it on planet Earth, basically this DNA-based life form. The fact that at the core, life is digital. Basically, if you look at DNA, it is a digital medium that encodes digital information in not 2 bits of information, but 4 letters, not just 1 bit of information, but 2 bits of information with 4 letters at every position. So it's not zeros and ones, it's ACGT, but it is digital. And I think that type of information is fundamentally different from, say, the equations that you need to describe, I don't know, differential equation systems or fluxes and, weather patterns, et cetera, and sort of gravitational pulls and, gravitational waves, et cetera. So the combinatoric nature of digital information, the fact that, with 3 billion letters, you have 2 to the, or at least 4 to the 3 billion, possibilities. And that, I think, is, perhaps more information rich than almost anything else in the universe. Like, basically, if you look at how much information is encoded in these 2 meters worth of DNA inside each one of your cells, it is truly mind-boggling to sort of, fathom that you truly have enough bits of information there to describe, galaxies and, entire cosmos, basically, in every one person on the planet. Now, there's the transformation of this information through the birth of every new human being and, the conception of every new zygote that start dividing and now has a body plan. And there's such a beauty in this automaton that is life, in this sort of self-contained cellular machine, which has so many properties that engineering systems would only dream about. The ability to self-replicate, the ability to metabolize, to basically take materials from the environment and just make them its own. I mean, think of R2D2 basically just like picking up a bunch of screws and picking up some scraped metals and just reproducing its own organs. I mean, this is an incredible feat of engineering. The fact that we can metabolize, we can sort of take other life forms like, a piece of celery and carrots and, fish and, all kinds of beans and, meat, and just, digest them down and make them our own. Like, just like if we could make robots that do that, we would be, thinking of ourselves as gods, basically, to sort of be able to create that. So that there's that aspect of metabolism. There's the aspect of reproduction, of every cell being able to divide. Like if you look at monozygotic twins, they're a single cell that basically splits. And from that one cell, you basically get 2 identical human beings. So one cell would have made a human, 2 cells make 2 humans. And, there are experiments where you basically can take the early cells of an embryo of 2 mice, for example, and then blend them, and somehow they will reorganize and form a single mouse. So it's truly magnificent that you basically have this ability to, number one, divide, and all of the information is encoded in every one of the 2 parts that have now divided from the 2 daughter cells. But also the developmental body plan that is encoded in just a small number of genes. The fact that I know where the anterior and the posterior is, the dorsal and the ventral. I know where the limbs, begin and end. I have a fully formed irrigation system that channels blood through all of my cells to nourish them, a fully, integrated immune system and circulatory system and, sort of musculature and the bone structure, et cetera. Like if you basically just think that every one of those cells in your body are in fact exactly the same information, the same exact 3.2 billion letters are basically reused by the same automaton to now make all of these self-connected, intricately intertwined systems of communication within our bodies. That's unfathomable. And again, I can't even begin to describe the next level of complexity, which is probably the most complex system in the universe, beyond even the human genome, which is the human brain. So the fact that that same set of genes not only encodes a fully functional body plan, but it encodes an incredibly malleable cognitive system that is able to process information at a speed so much faster than the time scale of transcription and translation and all of the basic cellular processes that work inside our bodies. So basically, you can think of the vehicle as the body, and then this incredible sort of cognitive system encoded within our brains that somehow has managed to have millisecond reactions to a much faster moving world around us, despite the fact that transcription takes minutes and sometimes hours for, some of our genes. So we basically have many different timescales of layers of processing through which this single unit of information has now encoded layers upon layers of interconnected systems, one of which is sufficiently complex to not only comprehend the cosmos, but go and explore it and build machines that go to Mars and, travel outside our solar system and, image our Milky Way and detect gravitational waves and sort of learn about ultrasound and things that our bodies never evolved to capture, like infrared and ultraviolet, and sort of expanding our senses, expanding our ability to sort of comprehend time and space from the ultra small down to the quarks to the ultra big to the edge of our universe, from the beginning of time with the Big Bang and beyond, to the end of time of sort of how our galaxy falls apart and how our sun implodes and how all of the universe ends. I mean, to me, the fact that within a lifetime you can learn about the whole beginning and ending of the universe, from the micro small to the ultra big, etc., thanks to this cognitive system that you have between your ears, is magical. It puts humans and planet Earth in such a special place in the universe, because the universe is kind of boring and life on Earth makes it exciting. And this concentration of information encoded within one human cell by itself brings order and meaning and, beauty to the universe. Like, who's there to appreciate it if it wasn't for life? Like, we would have these most beautiful sunsets on Earth and no one would be around to sort of appreciate them and appreciate the beauty of our galaxy and the cosmos if it wasn't for life. And, yes, animals can appreciate them and that's life and that's a magical thing, but humans can appreciate them at such a deeper level. So, and again, I've only talked about the information in one human genome, and now you can think of the information in all of life on Earth, like from the viruses that constantly attack us to the bacteria that have taught us how to defend us, against them, and how to edit genomes, through CRISPR, and to the, wealth of plant life and, mushrooms and all of these, sort of kingdoms that are surrounding us, to sort of marine life and, the quote unquote boring tetrapods that exited, and, this incredible wealth of inventions. So if you look at the history of evolution, there are gazillions of life forms and molecular machines that are constantly invented through the bacterial kingdom, and that are sort of reused and dispersed through the evolution of life, that are teaching us about chemical reactions that we never imagined possible, about enzymatic sort of metabolic changes, about drugs, about, defense mechanisms that are just so intricate. And life is constantly evolving all of these things, surrounding us. A tiny little snippet of which are the 20,000 genes, at least protein coding genes of the human genome, and maybe another 20,000 non-coding genes, which, and sort of 2.3 million regulatory elements that sort of make up human circuitry. But that's such a small, tiny part of the diversity of, biological inventions. So when you think about sort of information in DNA, you can't help but marvel at how little we have explored, and how that little thing inside, each one of our cells listening to this podcast is so incredibly rich, but also how much of a tiny little part it makes of the amazing biodiversity of planet Earth.
Yeah, that's fascinating. So, I mean, of course, when we look at from physicists' point of view, there is this, again, increase in complexity, as you already explained, and of course, the life form is the most complex thing in the universe. Um, so when we are talking about this complexity, we can also talk about this second law of thermodynamics, which basically explains the increase in entropy. That means um, anything, or with the time, disorder increases. But what happens, I mean, this is also clear that life is basically sort of against that law, because it, it explains the order coming from disorder. But what happens in the case of genome, or in the case of information, that once we go into more complexity, more complex form of life, what happens to the organization? What happens to the kind of disorder? Is it more disordered, or it's more ordered in a way?
So you can basically look at life in the form of gradients. I basically, I think building a proton gradient in a membrane, for example, if you take the mitochondrial membrane and sort of the buildup of energy through the separation of protons, I mean, that's, fundamentally the basic building block of, all of life, of basically fighting against entropy, of sort of increasing separation. Things don't just diffuse, but the membrane and the ability to sort of channel things. If you think about Maxwell's demon of sort of pushing molecules, in one side of a membrane, that's basically a fundamental building block of all of life. And one word to summarize it would be compartmentalization. The fact that you build compartments, that the cell itself is separating its self, the notion of a self, from the non-self, from the outside. The fact that there's a fundamental difference upon which, for example, metabolism is based. So metabolism is taking the non-self and using it to make more of the self. And you basically have this ability to create this concentration of nutrients, to increase the concentration of nutrients, to sort of constantly push things in rather than sort of let them diffuse out. And this is unmistakable for life, for basically the sort of basic building block of life. The fact that you're able to sort of fight back against entropy, to sort of increase order rather than disorder. Now, if you think about sort of prokaryotes versus eukaryotes, even at the level of a single cell, a eukaryotic cell is much more compartmentalized. You basically have these energy producing molecules, or sort of compartments, not molecules, that are your mitochondria. So basically they're an ancient organism that has still, remnants of its own DNA. So there are 13 genes that have still been maintained from that ancient engulfment event where we basically ate up a mitochondrion. We have a nucleus, which basically compartmentalizes the DNA storage of information from the cytoplasm and all of the sort of cellular activities. Even within the nucleus, you have dozens of compartments. You have the nucleolus, for example. You have, all of these different organelles, even within the nucleus. And of course, in the cytoplasm, you have chloroplast, for example, for, plants. You have, the endoplasmic reticulum. You have, the cytoskeleton. You have all of these different cellular compartments that sort of have increased this compartmentalization. So I would say that, there's of course a lot more complexity, but there's a lot more ordering to make that complexity function. And if you look at chromatin organization, if you look at sort of the topologically associated domains of our chromosomes, there's just so much modularity and hierarchical organization that allows you to now organize these increasingly complex amounts of information into sort of manageable parts, where you can sort of open up the necessary compartments for every cell type. I mean, I was describing earlier how each one of our cells has exactly the same DNA to a large approximation, and yet looks completely different from each other. How is that possible? That's possible through this compartmentalization. It's possible through the modularity. It's possible through the fact that even though all of these cells have exactly the same DNA, or at least, a subset of the 3.2 billion bases that every cell starts out with, there's this different subset of that is being used in any one of these cell types. And the morphology of these cell types is dramatically different. If you look at the shape of neurons, you basically have, every one of our cells is like 1,000th of the width of a hair. These are tiny, tiny, tiny little cells. And yet axons, for example, from our neurons can stretch all the way from our brains to our toes with a single neuronal connection. You basically have this, incredibly elongated cell alongside your microglia, for example, that are, functioning as immune cells of the brain, as sort of, cleaning up everything inside the brain, as your retina cells, or the lens of your, eyes, or the, follicles of your hair. And, you have this incredibly diverse, like your muscles, basically the ability to sort of be able to move your body comes from these multinucleated, elongated cells that are able to sort of use these stress, attachments to the bones and to your cartilage. And I mean, it's just incredible, the specialization that you have to make the human body function. So as you start thinking about sort of, information and the fight against entropy, I would argue that the more complex you are, the more entropy you kind of have to fight against, and the more compartmentalization you end up with, and the more organization you end up with. I mean, it's rule number one of any kind of systems engineering course, the fact that in order to achieve complexity, you have to have modularity. The concept of abstraction layers is probably the most foundational concept of computer science. The fact that you can abstract away the bottom layer and only worry about your layer, and then provide an interface to the layer above, etc. So I think we see a lot of that in, biology, in the sort of the organization of those cells. Basically, they're all based on quantum physics at the limit. And then, you basically have abstraction layers upon abstraction layers of obscuring that complexity to build upon manageable units and layers of information.
So does that mean, for example, in the unicellular organisms, it'll be easier to read the information than like reading that information in the eukaryotes or in more complex organisms?
I think the gene regulatory circuitry of bacteria is a lot simpler than the gene regulatory circuitry of, say, human. So you basically have a much smaller number of regulators, a much smaller intergenic space, so much more compactness there, a smaller set of regulatory programs. You have operads with, sort of many genes encoded in a polycistronic way from the same transcript. You basically have, just a small number of modules, if you wish, whereas eukaryotic genes, for example, have many, many, many more regulatory elements. They can be millions of nucleotides away, looping together. that allows them to have different sets of regulatory elements for every cell type where that gene will be utilized. You have exons and introns and splicing, which allows you to sort of break the gene into smaller modules of domains that can be mixed and matched together to create enormously higher protein complexity from the same number of genes. So basically you can have 20,000 genes, but the number of proteins is in the hundreds of thousands of proteins that you can make from these small number of genes by mixing and matching these domains through different gene regulatory elements that act pre-transcriptionally and post-transcriptionally through the splicing level to basically create units of function that are, much more diverse. So absolutely, you basically have this exponential increase in the complexity through these layers of regulation that have evolved compared to the much simpler set of regulatory principles of bacteria. I mean, again, the principles are fundamentally the same, but you just have layers upon layers upon layers of additional added complexity to deal with the huge information expansion.
Yeah, and so basically, you have already kind of described the reading information means like when the information is stored in DNA, it simply gets read in the form of RNA and then gets translated into the proteins. So the interesting thing here is in the year 2001, when the first human genome was published, and in 2004, basically you started your tenure. So fresh information, and I think that's the point where also the scientists were shocked, because they were thinking that there'll be more number of genes in humans, for example, compared to the other species, but that wasn't the case, right? Do you remember some stories around that time about the human genome?
I was there in Washington, D.C. with Francis Collins and
Wow.
Eric Lander and, all of the celebrations that happened, for the completion of the human genome. And I have a figure that I made that's in the human genome paper. So if you search for Hox genes, I actually made one of the figures in this massive paper, basically showing how repeat elements are actually depleted in the developmental Hox clusters. I lived through that time of basically watching the first assembly of the public human genome in Serafim Batsoglu's computer, and being enthralled not by the assembly program, but the fact that Serafim had a text file where he had pages upon pages upon pages of ACGT, and I'm like, that is my life's mission, like understanding that code. And I was very fortunate to again be at the right place at the right time. Basically, the tools that I developed during my PhD for understanding the content of genomes through their evolutionary signatures have actually become the foundation upon which the current protein coding gene set of the human genome is built. So when we look at the set of protein coding genes in the human genome, my work and my lab have been, a key part for that. So we've been part of the GenCode project, the, sort of gene set of the human genome, basically developing methods like phylo-CSF, or codon substitution frequency, and phylogenetics, that basically tells us whether a particular segment of the DNA is selected evolutionarily for a protein coding function. And that is at the foundation of what is the gene set of humanity. So where are all the genes? The reason why we know that there aren't hundreds of thousands of genes is because of programs that, Chris Burge initially through GenScan, and then, my group through phylo-CSF, and, many other groups have basically made to look for the signatures initially in a single genome based on the splicing motifs and the sort of codon frequencies and triplet, nucleotide frequencies of coding versus non-coding regions. And then ultimately through the comparative genomics of initially 4 species of mammals, then 29 species of mammals, now 200 species of mammals, that basically allow us to look for how different segments evolve in order to then figure out which ones are protein coding and which ones are non-coding. So it's very humbling to be part of this mission for understanding the basic building blocks of life, and human life in particular. So Mike Lin initially, and then Erwin Youngrice in my lab have basically sort of carried out this operation for more than 20 years, first building the basic signatures and then using them to understand the genome. So and that's one small part of the genome. That's 1.5% of the human genome. That's it. The protein coding parts, 1.5%. Then there's the 98.5% to understand. That's much harder. So basically what my group has also done is initially use comparative genomics to discover the dictionary of all regulatory motifs based on their genome-wide conservation patterns, based on the ability to sort of recognize that the same pattern of TCA spaced by 10 nucleotides from GCA is in fact repeatedly conserved in thousands of places in the genome. So that allowed us to now build a basic dictionary of regulatory elements in the human genome. We then developed methods that read the epigenome, that read the very basic, the same exact basic foundational tools that the cell uses to remember which regions are important for its function, to remember that it's a neuron and not a liver cell and not a heart, smooth muscle or other kind of cell, that or a vessel, and so on and so forth. Basically, all of these cell types remember what cell type they are through specific modifications on the packaging of the DNA. So the DNA is wrapped around these histone proteins that make up nucleosomes, and every 200 nucleotide chunks has a set of modifications that encode what is in that package, so that when you're searching for information in the human genome, you don't have to read through all 3.2 billion letters, but you can sort of say, okay, well, in this cell type, these are the active regulatory regions. So when I need to turn on genes, I'm going to only search through the subset of the genome that's, active in that one cell type. So we've basically developed methods to not only use comparative genomics and all of the sequence pattern discovery, but to also use epigenomics and to read that information to understand where are the building blocks of gene regulation, where are the control elements of human cells. And we've now done this across hundreds of cell types. We had a paper in Nature on the 20th anniversary, actually, of the human genome that presented EpiMap, the most complete yet annotation of the human epigenome, that basically allowed us to, define the activity patterns of 2.3 million regulatory elements across 830 cell types and tissues in the human body, and how they turn on and off, how they're connected to nearby genes, how they're connected to their upstream regulators, effectively understanding that circuitry, which now allows us to go and understand not just the gene content, but also the non-coding content, and eventually understand how disease manifests through genetic variants that predispose us to different diseases through infinitesimally small effects, to basically then understand how these effects are manifesting through these circuits that we have uncovered. So being able to understand these circuits allows us to now take non-coding variants across thousands of genetically associated regions with human disease, and then understand who are the upstream regulators that bind these non-coding variants, these letter differences from one human to another human that might predispose us, ever so slightly for cancer or Alzheimer's or obesity, and then go and understand who are the upstream regulators, what are the downstream target genes, what are the motifs that are disrupted, what cell types do they act in, what biological pathways are they a part of, so that we can then have a rational approach to designing new therapeutics, to sort of understand how we can use these to transform medicine, to transform the way that we go about reversing that circuitry in order to alleviate the burden of disease for every person in the planet, regardless of whether they carry those variants, to basically understand through the people who carry the variants, the basic circuitry. But once we understand the circuitry, build drugs for everyone that utilize these circuits and manipulate these circuits, regardless of the genetic diversity of each person.
Fascinating work. So one quick question that I had was, so what is the meaning when we say that we are 98% similar to chimpanzees or bonobos, for example?
There's very, very few things that have changed in the 5 million years that separate us from chimps. So basically, humans and chimps diverge from a common ancestor. You know, the chimp lineage went one way and made them more chimp-like, and the human lineage went another way and made them more human-like. There's dramatic differences if you look at the physiology of humans. You know, we are the naked ape. We don't have a lot of hair. We also have this giant expansion of the neocortex. Basically, you know, the chimp neocortex ends here. Like this whole part is humans. So it's quite remarkable that, you know, we have so much more cognitive capabilities, and there's specializations in the types of neurons. There's specializations in the circuits that are formed. There's specializations in the early life learning, the fact that our brain continues to be extremely malleable through our youth, all the way to 18 or so. Our brain is just extremely malleable. That's a period of evolution, of development that you don't see in the other primates. You basically, that's something unique to humans. So if you look at biology, we are amazingly different, especially when it comes to cognition. But if you look at how many differences does it take genomically to make that, very, very few. All you need is, you know, for one gene, which is, say, the regulator of the timing for how long will the cells keep dividing in your brain. All you need is for that one regulator to be slightly more expressed, or for, you know, these regulatory elements to be, you know, slightly more numerous. And suddenly you go from sort of having very few neurons to having a huge number of neurons. So if you look at the number of DNA differences, you might just need one to basically have an enormously larger brain. So, you know, we shouldn't sort of count the differences in just simply nucleotides, right? Because basically, if you go in as an engineer and you try to sort of, you know, change an engineering design, so from this automaton, which is programmed, suppose that you've written the code, like, you know, you have for your little like robot, like go right, you know, go straight, turn right, go straight, you know, put the pen down, you know, then start drawing a circle, etc. To make a bigger circle, you just need to change one letter. It basically says, you know, instead of going 3 centimeters, you go 30 centimeters, right? It just takes one letter. Once you have the whole robot built, all you need to do is change one letter for the program. But of course, biology doesn't work that way. It doesn't just change one letter, it changes millions of letters. So, you know, if we say, oh yeah, well, humans are 99.9% identical to each other. You and I are 99.9% identical. I mean, we are basically identical. But if you look at the 3 billion letters of the human DNA, there's still millions of differences. We have 3 million differences between any one of us, you know, any two of us. So basically, any one person carries 6 million common variants that are different, and, you know, thousands of rare variants that are different from, you know, other humans. So with chimp, you know, it's a lot more, but not that much. And yet, you know, even just a small number would have been sufficient. And these large numbers are basically giving us all of the other functions that are needed to, for example, support our much larger brain. So, our brain is only 3 pounds and it consumes about 20% of our energy. 15 to 20% of our energy goes to just 3 pounds of goo in between our ears, under our skull. So you need massive metabolic changes to make that possible. You need massive changes in the irrigation of, these frontal cortex, of, all of these neocortical regions, in the circuitry and the connectivity between all of these ancient brain regions and these new regions. The ability of our cortex to now control so much more of our bodily functions, of our, reasoning and thinking and memory and spatial organization of sort of how we remember events and places, etc. I mean, all of these things are, of course, highly interconnected with each other. So it's not just enough to just have more neurons. You need all of the circuitry and the wiring and the, sort of energetic needs and the feeding and the clearing out of debris. When you think about Alzheimer's, why do humans get Alzheimer's and animals don't get Alzheimer's? Probably because we use our brain to such greater capabilities. Basically, we push our cognition to such an extreme as a sort of super high performance machine. We're not just like, good old, I don't know, beat up pickup truck, rolling down the highway. No, we're a race car, running at the limits of what the physics will allow it to do. And, there's some compromises that need to be made in order to sort of maintain that machine. And some of those compromises have to do with the clearing of debris, with, sort of optimization for how long of a lifespan will I have a functional brain for, and, all kinds of other things. So you basically have to understand the human-chimp divergence in the context of all of the functional specializations, which are enabled through this, 3 billion years of getting good at evolving, through the modularity, through the layers of organization, through the hierarchical construct, so that when you have mutations in the DNA, you don't end up with an inside-out person. No, you end up with slightly bigger brain or slightly longer limbs or slightly, sturdier column, and so forth, like spine, and so on and so forth. So basically, these are all changes that are still biologically interpretable in a fully functional organism. I mean, that's what's amazing about biology. The fact that if I took your hard drive and I bombarded it with 1,000 different bits altered, after the first 10 bits, it would likely just crash and malfunction. Like, I won't boot now because I flipped 10 bits in your computer. In humans, you can flip thousands of bits. You can flip 6 million bits between any two people. And yet you get a perfectly valid human being, perfectly functional human being. I mean, you and I have 6 million differences, and yet both of us have, ears and eyes and noses and, neurons and, like all of these things that are still functioning despite this incredible diversity. You see what I mean, right? It's not just like I get a longer nose and suddenly the bone is going to be sticking out of the flesh. No, it's still a fully formed nose. And, if it's longer, it comes with all kinds of auxiliary things that sort of make that nose fully functional.
Wow. Yeah. So here, let's say if we kind of recollect everything. So with the increase in complexity in biology, of course, we see that the genome size increases, but it's not really the number of genes that they increase. That they are somewhat similar. It's basically the known coding region, which basically means that it doesn't produce any protein, right? That is what which expands, right?
That's exactly right. It's not the basic building blocks of proteins. You don't have new proteins. You don't have a new brain protein that will basically suddenly make you smarter. No, you just have the same building blocks rearranged and controlled and expressed slightly differently. And that's sort of the beauty of evolution. The fact that, yes, sometimes there's dramatic events like the whole genome duplication, which allows you to have now 10,000 new genes that you can do anything you want with. And, you can specialize these genes. You can create new domains. You can sort of, every now and then, you can create new genes out of nowhere. Most of the time you get new genes by duplication, which allows you to sort of start tinkering with something that's already functional. But every now and then you get a frame shift where basically a stretch of nucleotides is translated in a completely different set of amino acids because you've now shifted everything by one. So, what read like a set of 20 amino acids is now read as a completely different set of 20 amino acids. So sometimes this will actually be beneficial and those amino acids will still be functional. And we have, dozens of regions in the human genome where one strand is translated into two different sequences. You have an amino acid sequence here and off by one, fully functional amino acid sequence there. And yes, there's 3 billion nucleotides of the human genome. And yes, 98.5 of it is non-coding. So you have plenty of real estate, but it's like your neighbor came and built a house in the middle of your house instead of just like an entire field in Arizona where there's like no houses whatsoever. And yet your neighbor comes and builds a house right on top of yours. So that's basically what happens in the human genome for some of these regions. The fact that, you can still have two fully functional proteins encoded from the same nucleotides. These are completely unrelated from each other. And of course, after a genome duplication event or a gene duplication event, you can now relieve some of that overlapping complexity. And one is now free to specialize with this new set of amino acids that he created. And the other one is free to take on the old function or a new function and so on and so forth. So this is sort of typically the way through which new complexity arises, but over very, very large timescales. In the timescale of the human-chimp divergent, you basically have almost no new protein coding genes. What you have is small differences in the circuitry that basically change functions dramatically.
Yes, and this non-coding regions, they are also kind of labeled as junk DNA. And this is where your work also comes in, whether it is junk or not, right?
Well, it's such a terrible word, but first of all, junk DNA doesn't mean garbage DNA. Junk has a connotation of, well, there's probably some good stuff there. It's like your junk. Sometimes you just, you go in the junk in your garage and you find all kinds of useful things. So at least it's not called garbage DNA. But even junk DNA has, this weird connotation. It's basically, again, 98.5% of the genome doesn't code for proteins. That doesn't mean it's not functional. It just means that it doesn't make proteins. There's plenty of it that makes non-coding genes that's still transcribed and makes a fully functional RNA that has all kinds of functions as RNA. There's plenty of it that controls these light switches that basically turn the genes on and off, and that sort of make the DNA fold up and recruit the regulators to start transcription and so on and so forth. There's plenty of it that serves functions as spacer because you need lots of room for all of these regulators to bind. There's lots of it that sort of has roles in attaching to the lamina of the nucleus for non-coding regions that are sort of very often heterochromatic, that basically are sort of stably repressed to be attaching there. And so you need enough linker space. You need space for, splicing events to happen. And of course, you also have repeat elements. What are these repeat elements? These are basically selfish, self-replicating, transposable elements whose only function is to replicate themselves. So basically you have, imagine that you can create that at some point in evolution arises a genomic element whose protein product excises its own element, its own DNA, and then copies it. That element will now propagate and will make thousands of copies of itself in the genome. So about 50% of the human genome can be traced to these ancestral repeat elements. So there's massive, massive amounts of human DNA that basically is due to these viruses, like these transposable elements, these self-replicators that just make more copies of themselves, purely selfishly. They don't care about us. They just care to replicate. And we have developed massive machinery to shut them down, to basically make sure that they don't get, expressed and sort of go replicating, because then they'll wreak havoc on our genome. But what's really interesting also is that these same elements that are purely selfish were co-opted by mammalian genomes to basically build a whole new layer of circuitry by exploiting these regulatory regions, because a self-replicating element has amazingly potent promoters and enhancers that basically make it turn on. Why? Because those elements that had slightly more potent ones were selected by self-replication, and then random mutations accumulated. The ones that did not replicate as well did not replicate, and the ones that replicated slightly better continued spreading. So they had more opportunities to accumulate additional mutations, some of which made them better replicators, some of them which made them worse. So over evolutionary time, these transposable elements were basically fodders for the evolution of new regulatory control regions, which are now split and scattered throughout our genome and can be co-opted for building a more complex regulatory machinery, such as what happened in the mammalian radiation when the asteroid in the north of Chichen Itza were basically, sort of wiped out the dinosaurs and created these evolutionary niches in the airs, underwater, and, on land for mammals to radiate, and, eventually primates, and eventually humans. So all of that was in part mediated by these self-replicating, purely selfish chromosomal elements.
And sorry, so how much time do we have? What's your...
I don't know. Let's take another 20 minutes, 30 minutes.
Okay, perfect. Yeah. So, so about these transposable elements, do you think that they may have something to do with the viruses in general? I mean, not only transposable elements, but it's also the non-coding DNA that is, whether just by series of infections, it gets added into different species. And that's why we see increase in the non-coding DNA.
Well, I would be tempted to just dive into more of your previous questions, because, I think we're going to go down a rabbit hole. I have a lot to say on that, but maybe,
okay.
Maybe you want to sort of go forward to your second question.
Do you ask, then maybe we can go to your work. Okay, yeah, so maybe we can talk a little bit more about this genome-wide association studies that your group is working on.
So as a system engineer, you can basically think of the best way to understand a machine by poking it, by sort of making minute little changes and then seeing how it functions. And a lot of genetic studies basically used this principle to understand the basic foundational building blocks of biology. They basically went into viruses, they went into bacteria, they went into unicellular eukaryotes, they went into model organism animals like the worm and the fly and the mouse and the zebrafish, to basically carry out perturbations and then see what happens. So there's two ways to understand genome function and sort of biological function. One way is to wipe it out, basically take a gene, knock it out, and then see what happens. The other way is to take that same gene and overexpress it and see what happens. So basically both loss of function and gain of function. And the biochemist way has been, let's take this protein out of its system and then see what it does. The geneticist is, let's take that protein inside its system, wipe it out, and then see what happens. So the changes that basically gave us all of the foundational biological knowledge with which we understand life, including, viruses, bacteria, eukaryotes, and ultimately, animals and humans, happen through these genetic studies of perturbations. Basically, scientists going in and perturbing individual genes and then seeing what happens. And that's how we learn about a lot of the basic cellular machinery, the basic replication, all of the functions that are shared at the sort of at the level of life. We learn from viruses and bacteria. At the level of eukaryotic life, we learn from yeast perturbations. And then from animal life, we learn from worm and fly and zebrafish and mouse. And I should say from mammalian life, mouse. So that changed fundamentally in the last 20 years. So since the sequencing of the human genome, we are finally able to understand biology in the human first, rather than, oh, we figured out something by knocking out a gene in yeast, and then eventually we figured out that the ortholog, that the corresponding gene in human, has probably a similar function. But what's really fundamentally different now is that humans have become the organism of discovery. How is that even possible? I mean, of course, you don't go and perturb humans, but you go and sequence humans. And you then figure out these minute differences between all of us. These 6 million letters that we were talking about of common genetic variation, which is inherited in every new offspring. You basically have the ability to study how these tiny little perturbations are in fact allowing you to result in tiny little phenotypic perturbations. So the challenge, of course, is that computationally, this is a much harder problem. If I knock out a gene completely in a mouse, you'll see that this one has no limbs, or this one has, a smaller brain or a bigger nose, or you name it. So you will basically be able to observe a dramatic difference. In humans, you don't have that. What you have instead is millions of tiny little differences, each of which might affect your height by less than 1 millimeter. How are you going to study that? The way that you study that is by measuring the height of hundreds of thousands of humans, and then looking at all of the differences in the DNA of those humans, and then noticing that people who have an A instead of a G at that position are on average 1 millimeter taller. It's crazy, right? Like when you go to bed, you are shorter than when you wake up. When you wake up, you are longer again. Your height changes during the day just because of gravity more than the vast majority of these genetic variants that we know are statistically unequivocally associated with a robust, statistically significant change in height, which is tiny. So that's where computer science comes in. That's where sort of using the latest and greatest in statistics, in machine learning, in algorithms, in sequencing and analysis allows you to now study the system through millions of tiny perturbations rather than one giant perturbation. What's the other challenge? The other challenge is that you don't get to study this one mutation in isolation. You can only study it in the context of millions of other mutations. So basically, every human doesn't carry one mutation. So in a mouse, you basically have an isogenic line, and the only difference between those two mice is that, boom, you've knocked out this gene. So number one, it's a big perturbation. Number two, it's the only change. With humans, you now have millions of changes all coming together. And that's where I think the most beautiful part of what we do comes in. The fact that we are able to study the system from a systems perspective. We're able to study these millions of variables through electronic health records, through single cell profiling of individual cells for their transcriptome, the set of RNAs that they expressed, and the epigenome, the set of regulatory regions that they have active for every single cell across thousands of humans. What does that allow us to do? It allows us to now start asking how these minute differences, which are robustly associated with phenotype, how are they acting at the molecular level, at the cellular level, at the organ level, at the epigenomic level, at the circuitry level, at the protein level, at the metabolic level, at the lipid level, etc. So basically, we're able to trace the path of causality of how genetic variants manifest themselves, where they manifest themselves, and how ultimately they lead to disease. And that, to me, has made the human into the model organism of the future and of the present. We are now able to learn more about mouse biology by studying the human than we learn about human biology by studying the mouse. It's amazing. The roles have been reversed. And the reason is because of computational advances, because of machine learning advances, because of the ability of genomics to gather massive amounts of data and to integrate it all together to understand disease. So that's what our group does. What we're basically doing is systematically understanding the mechanism through which these millions of genetic variants are leading to Alzheimer's disease, schizophrenia, obesity, Down syndrome, so many different disorders where we can now start studying like cardiac differences. So atherosclerosis, heart failure, differences in exercise response, differences in, so many different systems in cancer predisposition, in response to immunotherapy. We're basically studying all of these different systems through the same foundational principles. So in the same way that the genome led to a unification of biology, genome-wide association studies and all of these new systems genetics and systems genomics approaches are now allowing us to lead to a unification of disease, to basically understand for every disease, what are all of the systems across the whole body that are impacting the disease. If you think about Alzheimer's disease, you're like, oh, great, it's the brain. Yes, but the brain itself is extremely complex. You basically have a dozen different subtypes of microglia, 17 different subtypes of vascular cells, 30 different subtypes of neurons, of excitatory neurons, another 30 subtypes of inhibitory neurons, many different types of astrocytes and oligodendrocytes. So all of these different brain cell types are in fact slightly differently impacted by these genetic variants that are associated with Alzheimer's. And now if you look at the biological processes, they're found throughout the body. Metabolic processes, lipid metabolism, cholesterol biosynthesis, lipid transport, just to cite a tiny little number. And there's like suddenly correlations that we're finding between obesity and between vascular disease of the brain, so cerebrovascular, and between cardiovascular changes in the heart. So we're basically finding commonalities between all of these different systems that we're studying. Some students in my lab are studying metabolism, others are studying cardiac disorders, others are studying Alzheimer's disease. Turns out they're finding the same pathways are underlying all of these. So you can't just simply say, oh, I'm an Alzheimer's scientist. No, that's ridiculous. You can't be studying a single system at a time, because all of these systems are in fact interconnected deep down. So basically what's really happening in my lab now is that by working on all of these different systems, we're starting to understand the foundational principles of human disease, of how all of these different pathways, all of these different hallmarks of disease are in fact interconnected and sort of reused and rewired and recombined in every single disorder. We're basically finding that almost every complex trait that we're studying is connected to almost every other complex trait. And in the end, we're able to distill them down to a small set of biological mechanisms that we are hoping to develop drugs for. So one of the new collaborations that we have is designing new molecules using these graph neural networks and these sort of machine learning techniques for studying structures, for predicting which drugs will bind to what target genes. By using all of these networks to predict the new target gene that we should be going after, to predict the cell types where we should be targeting them, and then to collaborate with both computational and experimental scientists to design and synthesize these molecules to effectively enable rational therapeutics that go after all of these pathways, and that allow us to now have a toolkit, to have a combinatoric toolkit that allows us to now modulate the specific pathways that are dysregulated in every single person for every single disorder. That's the ultimate vision, to basically understand these commonalities, understand these basic foundational circuitry, and then use it to change medicine, to change human disease forever, to basically allow us to approach it from an engineering, first principles perspective.
Fascinating. Since you're doing so many things, I mean, there is no wonder, basically, your kids that they don't want to continue in this field, right?
I don't know.
Maybe you will address all the questions by the time they'll reach in the.
I doubt it. I doubt it. What we're basically doing is building a set of foundations, but upon these foundations, you can have decades and decades of work to crack open not just human disease, but also human biology, to sort of be able to start, addressing so many issues of equity, of, health, of enabling human diversity to thrive by addressing disease in a fundamental way, so that we don't have to exclude people like Stephen Hawking, for example, because they have ALS, but we can allow them to thrive, to basically use the amazing genetic diversity that has given them unique skills, in music, in literature, in dance, in, rhyming, in artistic abilities, or in, physics, in chemistry, in, literature and mathematics and computer science and, biology. Like, take the diversity of the human brain, take the diversity of talents and abilities and interests and drives and passions that humanity has, and not worry about the disease component, because we can address that. We can sort of engineer ways to make all of these diverse humans live a healthy and fulfilled life, but allow their creative juices to change all of human knowledge. And sort of that's what I think makes the human species unique, this accumulation of culture that leads to this exponential growth of ideas, of achievements, by being able to build across each other, by connecting all of humanity in this information superhighway that allows a discovery made in India today to have an impact in, Alabama and in, Japan and in Kenya and sort of, across the world. Everyone can access the same set of papers, the same set of preprints, the same set of Wikipedia articles, the same, books electronically for the whole world. And suddenly knowledge can accelerate so rapidly by allowing human diversity to thrive without the burden of disease, of sort of, holding these individuals back because of, either their genetic predispositions or their environmental shortcomings, et cetera. We have a long way to go, but the building blocks are being built now. And sort of, I really do believe that we are entering a new generation of medicine, that basically by understanding these biological principles, by understanding these foundations, by building these sets of hallmarks to understand disease and to understand every system and every organ in the body, we will then enable a new phase of humanity where we can sort of thrive in collaborative ways.
Yeah, I think I have two interesting things, or two interesting points to make here. So the first thing is that also this will allow people to kind of make changes in their lifestyle to prolong their aging simply. That's fascinating. The second thing that I see is that you have kind of unified the understanding of genome itself. Like, there is, I mean, of course, coding, non-coding, and whatever, we can divide the regions, etc. But like, from your understanding, from your work, I get that feeling already that you are looking at the whole genome as an information, and just looking at the points which are changing in the population, which is again amazing.
We are building on decades of knowledge. We're contributing our sort of small stepping stones for others to build on. But I think you need all of the above. You basically need genomicists that are taking the global approach, and then you still need the traditional biology, sort of take any one of these biological pathways and just push it to completion. And you need thousands of experiments for any one of these biological pathways to truly understand its marvelous complexity and functions. But yes, what we are contributing is a layer of building blocks and the sort of systematic association with disease to sort of predict the biological pathways, the genes, the circuits, the regulators, the target genes, the upstream controls, the downstream targets, and all of that as tools for the next generation of scientists to not have to work it all out painstakingly, experiment after experiment after experiment, to basically say, in my PhD, I already know this circuit. I'm going to go in and manipulate all of these different tools because I know where they are. It's not unexplored territory anymore. We basically now have the cartography of the circuits of the human genome, and we can use it for exploration. It's the difference between sort of walking down the savannah in uncharted territory and just using, Google Maps to sort of be able to know exactly where to navigate. And, sure, of course, in the savannah without a map, you can still make plenty of discoveries. But if you have the full cartography, then you can be much more precise about the types of experiments that you're going to carry out. And don't get me wrong, these experiments are absolutely needed. And you need both types of scientists to coexist, basically the sort of pathway-centered, and then the genomicists, the geneticists, the epigenomicists, the computational science, computational biology, the machine learning, to all coexist in a network, in an ecosystem of mutual respect and collaboration.
Yeah, as you said that as humans, we have this culture of sharing the information, and so that the next generation can build on something that we have already found. I think this is where we can talk about synthetic life, which is interesting, and whether understanding the circuitry of life can help us to also kind of create a synthetic life.
So again, it's very tempting for us to call ourselves gods by basically saying, oh, I typed in a genome, I put it in a cell, and out came a new species. Yeah, but you didn't start from scratch. You started from a cell, from a mother cell, and so on and so forth. So yes, I do believe in synthetic biology. Yes, I do believe that we can sort of build circuits that didn't exist before and sort of take parts from biology, not just from human biology, but from the immense diversity of viral, bacterial, archaeal, eukaryotic, animal, plant, marine, terrestrial, aerial, take all of these incredible diversity and mix and match components and circuits to basically build new building blocks, new circuits, new machines that allow us to sort of create a huge new diversity of functions. And then, that can have implications for nourishing the planet, for solving our energy crisis, for, addressing so many different foundational, societal, human level, planet level problems. And all of that is absolutely within reach thanks to this. So basically, our group is focused on understanding the human genome, but these techniques can be used to understand sort of the planetary genome, if you wish, to basically start building new synthetic circuits and co-opting them for other either sort of applications for engineering and for science, etc. But at some point, you might also think about co-opting them for human health. If you look at CRISPR, it's basically a bacterial defense system against viruses that we have now co-opted to edit human DNA. So who knows how many more of those discoveries remain to be made, where we can basically co-opt these biological systems in new synthetic circuits that can now allow us to improve human health, human cognition, human longevity, human resilience, et cetera, et cetera, et cetera. So I am very optimistic about our engineering ability to co-opt all of these incredibly diverse machines, but we should be very humble about it, because what we're doing is using the products of evolution to basically transform medicine, to transform, food, energy, et cetera, et cetera, et cetera. So, new biomaterials, new synthetic limbs, new human-machine interfaces, all of these things will be possible by exploring, understanding, and reusing the products of nature. The next stage beyond that is to now start designing machines, to basically design, perhaps using an expanded set of amino acids, perhaps using an expanded genetic code, perhaps using an expanded set of, protein, lipid, glycan interfaces that has never been invented, in nature yet, but that we as engineers can invent and then push beyond just the reuse and the rewiring and the sort of remixing and matching of these biological components to truly design new components and sort of create new synthetic building blocks. And, this will undoubtedly come as well as we get better about predicting protein structures, predicting chemical structures, etc. We can basically start perhaps designing new machines that were previously not yet invented by life.
Yeah. And then what do you think about the artificial intelligence? So as you were explaining that in the genome or in the life, we see layers upon layers acting, right? So is it possible that once or at some point we can understand how these layers are really connected? And if we have some sort of machine learning program which can just take on these layers and kind of start working or start creating those kind of phenotypes that we want to see, or somehow if we can kind of make it learn from the biological system and to see whether it can act on the instructions that we are giving.
I think that machines and humans will be working side by side and already are. So basically, if you look forward, it's crazy to imagine humans doing everything. No, I think that already machine learning and AI are part of our assistants. We basically, as humans, can design a process where 3 different steps at different parts of that process are in fact machine intelligence. And that's perfectly fine. And that's sort of how I see the future. I don't see machines working in isolation saying, oh, we don't need you humans anymore. We're going to do the whole thing. But I can imagine sort of, engineers working side by side with artificial intelligence systems that can solve sub problems amazingly well, much better than any human designer would be able to make, because they're able to sort of explore a much larger design space, sometimes come up with solutions that we humans wouldn't have thought of, or that actually are counter to many of the design principles of sort of how humans design systems. I mean, if you look at AlphaGo and if you look at, the chess, you basically have machines that are sort of coming up with strategies for the game that make humans go back and say, whoa, this made no sense. No human player would have done this move in Go. In the history of humanity, we haven't ever come up with such an idea. But now you can look at what did the machine come up with, and then come back and write a new book for how to play Go based on what we saw the machine doing. And we as humans can start reasoning about it and perhaps come up with new principles that will then let the machines come up with the next generation of solutions and sort of go back and forth. So I expect the same thing will be happening in sort of synthetic biology and in so many different disciplines, where by having humans and machines work side by side, you can sort of learn from each other. You can initially teach the machine based on a lot of examples, eventually let the machine figure things out by itself, and then continue creating. Look at the creations, learn more engineering principles, perhaps push it down a different path, and sort of this coexistence of human intelligence and machine intelligence, I think is not just the way of the future, but it's probably already the way of the present for so many different disciplines, and only more to come forward.
Yeah. So what do you think of the that what can be the biggest discovery of 21st century, if you'll predict?
I think it's basically bringing all these ideas and directions that I've sort of mentioned as our goals, bringing them to life. Basically, it'll take, many people, not just my group, but many people to make this happen. But having a set of molecules, having a set of drugs, having a set of manipulations for every single pathway in the human genome, to basically be able to rewire any aspect of our biology. On one hand, to combat disease, on the other hand, to extend the lifespan, and thirdly, to sort of perhaps improve the human condition, perhaps improve, our well-being, perhaps improve our cognition, perhaps improve our athletic abilities through systems that already exist in nature, but have not yet been co-opted by humans, and maybe have not been co-opted by the same human all at the same time. There's extraordinary abilities if you look at human diversity, and being able to find the right combination of molecules that allow us to manipulate these circuits to perhaps enable just a more complete human life for, so many people in the planet would be, perhaps the most foundational and dramatic change of the human condition that we have seen in the history of humanity. And that's just for human life. Now, the sort of whole human machine interfaces and working alongside AI and sort of, intelligent systems. Again, we've seen more advances in AI in the last 15 years than we have in, the history of AI and of course, obviously the history of humanity. So I think we are so close to that singularity. I think we're so close to this exponential growth in advances that, will sort of transform life as we know it. So I think within our lifetimes, we're going to see a world that our children will, couldn't have even imagined, and that we as children couldn't have even imagined. So I'm very optimistic, and I think it's going to be the right balance of containing our worst instincts and human flaws, and sort of embracing collaboration, coordination, and sharing of all of these advances with a much broader set of individuals than have traditionally been receiving those. And I think this comes from making advances that just make the pie bigger, that sort of allow well-being and health, and not just a healthy life, but a fulfilled life for so many more of us.
Yeah, since we are discussing about beauty of the genomes, what would you tell people, to read about, I don't know, genomes, if they want to work on genomes? What kind of message would you like to give? How do you look at this beauty in genomes?
I think it comes with an appreciation for the beauty of life, and for the beauty of the planet, and for the beauty of the animal kingdom, and for the beauty of the human body, and for the beauty of the human mind and the human brain. I think it sort of comes with humility, to being able to just observe, learn, appreciate, understand. And that's on the biological side. And then on the computational side, again, humbly sit down and, take classes in basic foundational statistics, mathematics, computer science, machine learning, artificial intelligence, sort of programming, build yourself a quantitative set of skills along with your biological and medical set of skills. And I think these are the two basic pillars upon which you can build a career of impact in that field. So and there's like a huge number of courses that are fully online. Sort of all of my courses are on YouTube. You can watch them freely. You can sort of learn from, Stanford, from MIT, from Harvard, from all of the universities in the world. You have also people who are not professors, who are just amazing communicators, who have put so much material out there for free. So I would say start with Wikipedia, start with YouTube, and this gives you the sort of entry point. And then you have so many more articles on, bioRxiv and medRxiv and arXiv for preprints, before they even go through peer review, for open source journals that are increasingly the norm, for, PubMed, and the, sort of forcing by NIH for all federally funded research to have open source preprints. that are sort of prior to the editors sort of having their input. So I think there's just incredible availability of breadth, but also incredible availability of depth for any one of these areas. And frankly, I wish I had 100 hours every day, because there's just so much to learn every single day that basically the most difficult part is finding the time to learn about all of these things that are now possible.
Yeah, fascinating. So with that, thank you so much for the conversation. Thank you for accepting the invitation. This was really good for me. I mean, I think I already told you that I saw you here in Brno, and after that, like once I listened to your presentation, I was following your lectures, your conversations on YouTube, and you've been an inspiration, and I think you will inspire more.
Thank you so much, Jitendra. Thank you for
Thank you.
doing this. Thanks for inviting me. And thank you for your service. Thanks for bringing all these awesome people to your podcast. I hope that this is going to continue to grow, and that sort of many people will be inspired and go into this field. We can't hire fast enough. We basically have a dire need for more computational biologists, for more computer scientists, for more biologists. So it's a discipline that's booming, expanding. And for all of you young listeners out there, please join our field, learn the foundations, and come join us for this mission to transform human life, human health, and hopefully all of biology.
Everything is for science. So thank you.
Thank you, Jitendra. Bye.
Bye.